Alright, let's break down a topic that sounds complicated but is actually super intuitive when you think about it. Gluconeogenesis. The name itself gives you a clue. Gluco for glucose, neo for new, and genesis for creation. So we're literally talking about creating new glucose. This isn't just some random backup system. It's a fundamental survival mechanism hardwired into our biology. Our bodies, especially our brains and red blood cells, are glucose hogs. They need a constant, steady supply of it to function. When we haven't eaten for a while, say overnight while we're sleeping or during a longer fast, our primary glucose stores, called glycogen, start to run low. That's when gluconeogenesis heroically steps in to save the day. So, why is this process so critical? Imagine you're pulling an all-nighter studying for your board exams. You haven't had a meal in hours. Your brain is firing on all cylinders, demanding energy. Without gluconeogenesis, your blood sugar would plummet, a state we call hypoglycemia. You'd feel weak, dizzy, and your cognitive function would tank. This process ensures that even when external food sources are unavailable, your body can maintain stable blood glucose levels. It's a brilliant internal manufacturing plant that keeps the lights on, ensuring your most vital organs are never left without their primary fuel source. It's what kept our ancestors alive during times of famine, and what keeps us going between meals today. The primary site for this incredible process is the liver, with a smaller contribution from the kidneys. The liver is like the master chemist of the body, constantly monitoring and adjusting metabolic pathways to maintain homeostasis. For gluconeogenesis, it takes non-carbohydrate precursors, think of them as raw materials, and skillfully converts them into glucose. What are these raw materials? We're talking about lactate, which builds up in our muscles during intense exercise, amino acids, particularly alanine, which come from muscle protein breakdown during fasting, and glycerol, the backbone of triglycerides released from our fat stores. Your body is incredibly resourceful, recycling these byproducts to create something vital. Understanding this pathway is non-negotiable for any future physician. It's not just about memorizing steps, it's about appreciating the elegant logic of human physiology. It's a cornerstone of metabolism that connects carbohydrate, protein, and fat pathways. When you grasp how the body makes its own sugar, you start to understand the pathophysiology of major diseases like diabetes, where this process can go into overdrive. You also begin to see why certain dietary approaches, like ketogenic diets, have such a profound impact on our metabolism. It's a core concept that will come up again and again, from endocrinology to emergency medicine. Now, let's get into the nitty-gritty. Gluconeogenesis isn't just the reverse of glycolysis, the process of breaking down glucose. If it were, it would be a futile cycle, creating and breaking down glucose with no net gain. Instead, it's a clever workaround. Most of the steps in glycolysis are reversible, but there are 3 key irreversible steps. To bypass these roadblocks, gluconeogenesis employs 4 unique high-yield enzymes that you absolutely need to know for your exams. These enzymes are the special forces of this pathway, making the seemingly impossible possible. They are the stars of the show, and their regulation is where the magic really happens. The first major hurdle is converting pyruvate back to phosphenylpyruvate, or PE. This can't be done in a single step. First, the enzyme pyruvate carboxylase, which is found in the mitochondria, converts pyruvate to oxaloacetate. This step requires biotin as a cofactor, a classic board question. Then, oxaloacetate is shuttled out to the cytoplasm, where the second key enzyme, PEP carboxykinase, or PPAC, converts it into PEP. Think of these 2 enzymes as a tag team, working together to bypass the irreversible pyruvate kinase step of glycolysis. This 2-step process is the official start of the gluconeogenic pathway and is a major point of regulation. The next irreversible step to overcome is the conversion of fructose 1,6-bisphosphate to fructose 6-phosphate. The enzyme that handles this is fructose 1,6-bisphosphatase. This is a simple but crucial step. It essentially just chops off a phosphate group, bypassing the phosphofructokinase 1, or PFK1, step from glycolysis. This enzyme is a critical regulatory point. Its activity is inhibited by substances that signal high energy, like AMP and fructose 2,6-bisphosphate. We'll talk more about that last one because it's the most powerful regulator of this whole process. For now, just remember this enzyme as the gatekeeper for the middle part of the pathway. Finally, we arrive at the last unique enzyme, glucose 6-phosphatase. This enzyme is located in the endoplasmic reticulum of liver and kidney cells, but notably absent in muscle cells. This is a very high-yield fact. Its job is to convert glucose 6-phosphate into free glucose, which can then be released into the bloodstream to raise blood sugar levels. Because muscle cells lack this enzyme, they can store glycogen for their own use, but cannot release glucose to help the rest of the body. This makes the liver the primary altruistic organ, responsible for maintaining blood glucose for everyone else. Its presence is the final checkpoint before glucose can exit the factory. So we have the pathway and the enzymes. But how does the body know when to turn this factory on or off? The regulation is incredibly sophisticated, involving a beautiful interplay of hormones and local cellular signals. Think of it like a smart factory with a central command center. The two main hormones calling the shots are glucagon and insulin. These are the yin and yang of blood sugar control. Glucagon, released from the pancreas when blood sugar is low, is the go signal for gluconeogenesis. It tells the liver, hey, we're low on fuel. Start making some new glucose now. Glucagon doesn't just flip a single switch, it orchestrates a coordinated response. It binds to receptors on liver cells and initiates a signaling cascade that leads to the phosphorylation of key enzymes. More importantly, it stimulates the transcription of the genes that code for the key gluconeogenic enzymes, especially PEC. This means that during prolonged fasting, the liver not only activates the enzymes it already has, but also starts manufacturing more of them. This is a powerful long-term adaptation to ensure sustained glucose production. Cortisol, the stress hormone, also plays a synergistic role here, further upregulating these enzymes to prepare the body for a fight-or-flight response. On the flip side, we have insulin. Released by the pancreas after a meal when blood sugar is high, insulin is the stop signal. Its message to the liver is clear. We've got plenty of glucose coming in from food, shut down the internal factory. Insulin actively suppresses the expression of the genes for PEC, fructose-1, 6-bisphosphatase, and glucose-6-phosphatase. It effectively dismantles the gluconeogenic machinery at the genetic level, ensuring that the body doesn't waste energy making glucose when it's already abundant. This hormonal control is precise and essential for preventing dangerously high blood sugar levels, a condition known as hyperglycemia. Beyond hormones, there's also local control within the cell, known as allosteric regulation. This is like having supervisors on the factory floor making real-time adjustments. The most important allosteric regulator is a molecule called fructose-2, 6-bisphosphate. This molecule is a potent activator of glycolysis via PFK1 and a powerful inhibitor of gluconeogenesis via fructose-1, 6-bisphosphatase. Insulin promotes the formation of this molecule, while glucagon breaks it down. Another key regulator is acetyl-CoA, which builds up during fat breakdown. High levels of acetyl-CoA signal that fat is being used for energy, so it allosterically activates pyruvate carboxylase, pushing pyruvate towards gluconeogenesis to supply the brain with glucose. Understanding gluconeogenesis is not just an academic exercise, it's central to clinical medicine. This pathway is a major player in both health and disease. During periods of fasting, starvation, or even prolonged intense exercise, gluconeogenesis is our lifeline. It maintains the blood glucose necessary for our brain, which cannot use fatty acids for energy. In a healthy individual, this system works flawlessly. However, in disease states, particularly type 2 diabetes, this regulation goes haywire. In diabetic patients, the liver becomes resistant to insulin's stop signal. As a result, gluconeogenesis runs unchecked, even after a meal. This is a primary reason why diabetics have persistently high fasting blood glucose levels. The pathway's reliance on specific enzymes also means that genetic defects can have devastating consequences. A classic example you need to know for boards is von Gierke's disease, or glycogen storage disease type 1. This is caused by a deficiency in the final enzyme, glucose-6-phosphatase. Without this enzyme, the liver can perform all the steps of gluconeogenesis and glycogenolysis right up to the end, but it cannot release the final glucose product into the blood. This leads to severe fasting hypoglycemia, a buildup of glycogen in the liver, hepatomegaly, and an accumulation of precursors like lactate, causing lactic acidosis. It perfectly illustrates the critical importance of that one final step. Other clinical scenarios also highlight the importance of this pathway. For example, alcohol consumption can significantly impair gluconeogenesis. The metabolism of ethanol increases the ratio of NADH to NAD+ in the liver. This high NADH to NAD+ ratio inhibits gluconeogenesis by shunting its precursors, pyruvate and oxaloacetate, away from the pathway. This is why individuals who engage in heavy drinking without eating are at a high risk of developing severe hypoglycemia. It's a critical concept for emergency medicine, as a patient presenting with altered mental status could be suffering from alcohol-induced hypoglycemia, a true medical emergency. In summary, gluconeogenesis is a masterfully regulated and essential metabolic pathway. It's our body's defense against hypoglycemia, ensuring our brain and other vital tissues are never without fuel. Its regulation by insulin and glucagon is a cornerstone of endocrinology, and its dysregulation is central to the pathophysiology of diabetes. Furthermore, inherited defects in its key enzymes, like in von Gierke's disease, or metabolic interference from substances like alcohol, provide clear, high-yield clinical examples of its importance. Mastering this topic isn't just about passing an exam, it's about understanding the fundamental physiology that will guide your clinical decision-making for your entire career.